Hello, my name is Elizabeth Kohler, and I will be speaking to you about working towards a global solution to energy using lignin-degrading bacterial laccases. So just as a background, laccases are isolated from plants, fungi, bacteria, and insects. They are copper oxidases, and they use molecular oxygen to catalyze aromatic and non-aromatic compounds. And enzymatic reactions result in phenolic-free radicals and water. So we are particularly looking at bacterial laccases, and to date, only 3 bacterial laccases have been characterized and purified. And the benefit of using a bacterial laccase over other laccases is that they have high catalytic efficiency, the possibility of withstanding higher temperatures, low utility costs, and they're environmentally friendly since they only require molecular oxygen as a co-substrate and have water as a byproduct. Bacterial laccase that we are looking at is called PT-Lac, and it can be used to cleave lignin, which is part of a plant cell wall, into smaller soluble fragments to give access to hemicellulose and cellulose that can be fed to microbes to produce biofuels. An enzymatic assay was performed testing this, and this last tube we see here is lignin, a mediator, and PT-Lac. And the mediator is just a small molecule that aids in enzymatic oxidation, and the dark color of this tube is indicative of PT-Lac cleaving lignin into smaller soluble components. So to find the structure of PT-Lac, we prepared crystals at NSLS2 at Brookhaven National Lab using acoustic sample preparation. So we filled a source plate with crystallization conditions and protein, and also inserted a destination plate into the Echo 550. And the source plate moves over the transducer, which emits sound waves through the liquid, which produces as small as 2.5 nanoliter drops into the destination plate. On the right here, we have 2 destination plates that we used. This one here, just for size reference, each dot is 60 nanoliters, and each dot is a different crystallization solution with protein. So these are the crystals that we have achieved, we have obtained from PT-Lac. These first 2 here were early on, and they're polycrystals, so they diffracted very poorly. After some further refinement, we got nicer looking crystals that were also poorly diffracting because they are polycrystals, even though they might not look it. And then finally, after even more refinement, we were able to achieve these crystals here, which are very singular in nature. At the same time, we were crystallizing another bacterial laccase called PA5930, and from this protein, we were able to obtain very nice singular looking crystals that were well diffracting. So we were able to determine the crystal structures of both PT-Lac and PA5930. Here are the 2 structures. This is PT-Lac, this is PA5930. And the important characteristic of both of these structures is that they have the type 1 copper and the trinuclear cluster of coppers here and here. So substrates are catalyzed by the type 1 copper, and then the electrons are transferred to the trinuclear cluster where reduction of molecular oxygen and water take place. And the red dot in the middle of the trinuclear cluster is indicative of water. So one thing we were looking at is the coordination around the type 1 coppers. What is circled is the axial ligand, and it has been proposed in order to improve the redox potential of laccases to look at the axial ligand. So PT-Lac has a coordination of a histidine, histidine, cysteine, which PA5930 also has a histidine, histidine, cysteine, but PT-Lac has a leucine in its axial position, and PA 5930 has a methionine in its axial position. So we have proposed a few mutations of this axial ligand in order to improve the redoxability. So as mentioned, PT-LAC has many implications within the production of biofuels, and to further optimize the use of PT-LAC, we will study the mutations of the axial site around the type 1 copper, and we will mutate that to a phenylalanine, a methionine, and a tryptophan. And it is hypothesized that phenylalanine will increase the redox potential, while methionine will decrease the redox potential. And then for tryptophan, it is not a naturally occurring axial ligand, therefore it might not fit within the coordination, and the effects on the redox potential is unknown.